good example, let's say you want to increase sperm motility. And what you do is your test is you set up a little sperm racetrack and you have a microscope. Then you just cover the sperm with different drugs, different sperm, different drugs. You have a little race.
I'm Heather Grish, and this is the Wisdom of the Body podcast. This podcast explores the idea of body intelligence as the real key to learning the knowledge of life, or what we call Ayurveda in the ancient language of Sanskrit. We do that by connecting with today's creative leaders and experts who will help you listen to your body, trust your gut, and live in deeper harmony with nature. Come join me as we unlock the golden door to clear, direct perception and become very deep listeners. You can find the Wisdom of the Body podcast on Apple, Spotify, and Stitcher. You can also follow us by joining my email at heathergrish.com or on Instagram at heathergrish. I know you're going to love this podcast, so take a second right now to subscribe. Today, I'm here with Dave, a pharmacologist and biomedical scientist who I consult with when I see a client who's on a large list of drugs and I start to get concerned about drug interactions. He is going to talk with us today about drugs, and this is his second time on the Wisdom of the Body podcast. Thanks for joining us today, Dave.
My pleasure. Thanks for having me.
Is it really your pleasure, though?
It is my pleasure. I like talking to you.
Okay. Okay, so you said something in our first interview. We talked about drugs versus pharmaceuticals that are, I guess you could say, created by humans versus plants and things that are created by nature and the differences between them. We kind of started there. And you said something in our first interview that was kind of troubling to me. And so I
Only one thing?
Well, I wanted to talk about it again. So one of the things that you said was that all medicines are basically poisons. Am I correct that you said that?
I probably said something very close to that. I could be more precise in that definition, but that's essentially correct. There's definitely more precision that I should use. All medicines given are designed as specific poisons with the hopes to remove the disease process. And that's kind of a classical old school version of what a drug is. It's poisoning a process that you don't want to be working anyway inside the body. Something that's overactive, you want to reduce its activity, or something that's not active enough, you want to increase its activity, something to modulate the activity to put you back to remove the disease state. And you need to sometimes muck up the works. It's an old definition. It's actually a dictionary definition of the term poison. It's just not the most commonly used dictionary definition. Now, that is different than, let's say, replacement therapy, where you're taking insulin or something like that. That is not considered a poison per se. So anything, or giving growth hormone to somebody with dwarfism or something, those replacement hormones are not considered poisons. Or you're not poisoning a system. You're replacing something that's native to you anyway. You're just not producing enough of it. That poisoning can come in a variety of flavors. One is, of course, with a small molecule drug, but you can also poison with antibodies and things like that, where an antibody will block something and muck it up using an antibody. And that's becoming more and more in vogue to use in medicine, is to use antibody drugs to muck up some receptor. And it's more common when you're trying to affect what's called a protein-protein interaction. Protein-protein interactions are very common in signaling in the cell. And if you want to muck those up, very commonly, you need a much bigger kind of wrench. And an antibody is a very big wrench that can muck up processes like that.
Okay. Before we scare all the listeners away with you getting too technical.
Already.
Okay. So thank you so much for, starting to clarify what you meant in the last one. So now what I'm hearing is that there are different drugs that have different actions that essentially are modulating, and that poisoning is not what all drugs do, but that poisoning is what many drugs do. Is that correct?
Yeah, exactly. Also, I should let you know, there are definitely poisons in foods, which can cause issues as well. So there are notable foods like grapefruit juice, grape juice, and other types of juices that will poison certain enzymes, will stop, essentially not only stop, but permanently stop some enzymes in the gut. And that can create issues with other drug interactions and elevated drug levels if you're taking grape juice with some medicine that happens to be broken down by those enzymes. So poisons can come also just from normal foods that are you consider healthy. So potatoes have tons of carcinogens in them when you cook them, but our bodies are designed to tolerate a lot of these toxins and poisons, so we can tolerate some. But in the field of medicine and pharmacology, what you're doing is trying to exquisitely target a system to poison to make you feel better, to make you better. If you have a sense that that system has run amok, and there's several examples of an antibiotic is trying to poison a foreign bacteria in your body, trying to poison and kill them, as well as antivirals and anti-cancer drugs. Those are all, we're trying to selectively poison those things that have gone wrong inside our bodies and keep the healthy tissue spared.
Thank you. I really appreciate that. I think this word poisoning just seems really triggering for me. I don't know if anybody else had that the first time around. And I guess it sort of then begs the question, if something is considered a poison that could disable a process that's been going on that is actually harming us, and at what point something becomes a poison to where even that is not providing the benefit anymore.
Well, the most common effect in medicine when we poison a process, very, very often we're not as specific and pinpoint as we'd like to be. And you're poisoning some other process that we don't mean to poison, and you end up getting a side effect. And there can be really bad side effects, which is essentially poisoning some other part of the body, which we don't intend. And of course, if you read a label on a drug, you'll see a multitude of side effects. That's all from poisoning some other part that we don't mean to. And some of those side effects can be very, very bad. And there's a balance when a drug is approved that balances out what is the nature of the side effects versus what is the disease you're trying to improve. So if you're trying to remove acne, but then you've got a side effect of liver failure, well, that's never a tolerable balance between the level of side effects versus trying to clear acne, versus if you're doing an anti-cancer drug that will kill the person versus some problem with incontinence or something like that. Well, you'll always go for trying to cure the deadly cancer with incontinence as a bad side effect, but tolerable compared to survival. So there's always this question of balancing how targeted can we be in the poisoning and what are the considered off target effects. And that's always taken into consideration.
Right. I mean, it seems like I don't know of any drugs that don't have side effects listed for them, but I guess what are the decision processes? Are you aware of those decision processes when people are making drugs, of at what point the acceptance of the side effects is worth it for that particular drug?
Yeah, there's been drugs that have been removed from the market for really bad side effects. And it can be very, very sensitive in terms of the balance. So, one of the obvious ones was Vioxx, which was a Merck drug, and it was essentially given to millions of people. And the issues that can come up when you give a drug to a million people, what you'll never necessarily see in a clinical trial because you don't recruit a million people into a trial. So, if it's a rare side effect, but a really bad one, that can end up with the removal of a drug that could be even helping tens of thousands of people. So Vioxx was removed because several people had gotten a really bad illness, rare but bad, and died. And so it was removed because the actual drug itself, there were other alternatives. So the balance is very carefully weighed. And I will say, sadly, I believe the FDA in time has changed the value system a little bit. Safety is always a concern, but on the opposite side, they seem to be more in the frame of mind of if it's safe, I think the bar has been lowered a little bit for the efficacy part. There's a recent drug that's been approved for Alzheimer's. That's an example of this. It may be safe, but I think the FDA could be giving false hope to many, many people with Alzheimer's because this drug is not that efficacious. And that has changed, and that's like kind of the political level. So these balances, I think safety is always a major concern, and they're always very cautious. But the efficacy part, the bar, I think, has been lowered since 20 years ago. The bar was very high, it was very hard. But now I think there has been lobbying and complaining that the bar was set too high. And, there's people out there that have diseases that are in need and desperate, and you want to give some people hope. But I think preying on hope it just drains people of their pocketbook, frankly, and it doesn't really offer a lot of benefit. So yeah, it's still people are involved and politics are involved also. But I think that balance, and because of that, I think the balance on the safety side is always very strict, but the balance on the efficacy side to me has slipped a little bit.
Well, thank you so much for saying that. And you also mentioned earlier, hormonal treatments and how certain categories of medicines might be different, like not necessarily considered a poison, but hormonal treatment, since you mentioned insulin, and I don't think a lot of people are aware that that's actually a hormone. I think most average people seem to know that they have hormones, and women assume that it's like estrogen and progesterone. And if they looked into their fertility, then they've maybe looked at, AMH and FSH. But I think a lot of people probably aren't aware that insulin is actually a hormone. And how are hormones, I guess you're saying that they're essentially replaced? And are there instances in which hormones that we try to reduce them as well with pharmaceuticals that are synthetically made?
Well, a common one is if you want to change your gender, or there's also chemical castration. There are drugs that will suppress testosterone and estrogen and things like that. If you want to kind of change your identity, that's kind of a big thing these days. And that's extremely controversial, obviously. So, yeah, there are medicines that can alter hormonal levels in your body. And of course, the common one is the pill. That is a hormone analog, but then you're just trying to fool your body, essentially. You're just trying to fool it into thinking that it's not a good time to get pregnant. So let's tamp down the process that gets you pregnant. So there's ways to fool the body with natural analogs or analogs to hormones, and there's ways to suppress hormones altogether to try and shift things around. There's the different types of hormones, as you mentioned. So FSH and LH, those are also peptide hormones like insulin. And then there's the class of what's called steroid hormones, which are derivatives of cholesterol, and those are testosterone and estrogen. Estrogen being one enzymatic pathway process away from testosterone. This is why men who lift weights can get feminine qualities because they put too much testosterone in their body and it can get converted to estrogen. Whereas estrogen can't get converted back to testosterone. That's a one-way ticket. And then there's also called the glucocorticoids, and those are also derivatives of steroids, and they modulate things like your kidneys and the mineralocorticoids. So they're all different types of hormones, and they all have different purposes in the body and different targets. And in fact, there's communications that's called autocrine communication between cell to cell, very close by. So and there's paracrine communication. There's all sorts of different communication going on in the body. A lot of the hormones we're used to are systemically released, and different cells will kind of interpret that message differently. So there's a huge physiological treatise on the different types of hormones and the feedback systems involved.
I keep getting like triggered by some of the language that you're using, I have to be honest. So
Oh,
it's funny, like this idea of fooling the body, that we're trying to fool the body. And I know that there's probably a great intention behind it, but why would we need to fool a body?
free love. That's why we fool the body. Three is to be able to have sex without getting pregnant. It's taking a combination of estrogen and progesterone, and it alters your menstrual cycle and tries to eliminate ovulation. So if you eliminate ovulating, then you won't get pregnant. The only other time you wouldn't be ovulating is when you're pregnant. So when you're pregnant, you don't ovulate anymore. So it's kind of fooling the body into believing it's pregnant. So it doesn't need to, I mean, it's behaving as if you're pregnant, right? Because you're not ovulating. And normally, if you're a woman who has a menstrual cycle, you're ovulating every month, right? So if the pill stops that, so you may have a little bit of a period, but it's not the same as if you were not on the pill. It's different because there's no egg in it.
Yeah, apparently women don't ovulate in every menstrual cycle. I just found a study on that. There's actually a lot of them in which they're not ovulating, but yes, the pill does seem to shut off ovulation and trick the body into thinking it's pregnant is a really good example. Okay, so let's shift out of hormones because that's probably a whole separate episode that we could do together. I guess, since we're sort of talking about different kinds of drugs, and I'm coming at this from this idea of a lot of people wonder if things that are found in nature versus things that are made in a lab are different. And does the body like one of those more than the other? Is one of them better for the body than the other? This question sort of comes up a lot in the holistic health world and for people who are interested in that style of medicine. And I guess one of the questions I have is, are there any examples of pharmaceuticals that are made today where they're actually nutritive or promotive and they sort of build tissues?
Well, I mean, testosterone builds tissues. There's lots of any anabolic growth hormone would build tissues, but you only want to build tissues when you want to build tissues. If you're my age, you don't want to build tissues anymore. You just want to maintain your tissues.
Wait, why don't you want to build tissues anymore at your age?
Well, I mean, it's actually a very interesting question because it gets into a whole series of the sense of what aging is. A lot of what aging is as you get older and older, your body has been through many, many insults by living in the environment, including getting a suntan or eating lots of potatoes or eating lots of things that have little insults in them, or walking down the street and sucking in some exhaust fumes from a car, or living in industrial society. You're exposed constantly to environmental toxins and insults. And your body is always detoxifying in different ways. And every once in a while, your body just doesn't do a great job, or when a chemical escapes. And through time, through just lots of exposure, your body is always dodging these bullets. And one of the ways it dodges the bullets is it decides that a cell fate, one way to avoid getting cancer is to just turn off the cell's ability to divide. And as you age, you get more and more cells building up that have decided they don't divide. Now, you asked about growth. These are cells that won't respond to growth. In fact, they don't really behave as whatever the tissue of origin there is. If they're a liver cell or whatever, they're not behaving like a liver cell anymore. They're just kind of filler there. And they don't divide anymore, but they're not like helping the liver out anymore either. But they're not cancer, so that's good. But as you age, you accrue more and more of these types of cells. And there are people who believe that removing these cells that are just kind of hanging around, but they're using energy, but they're not like helping out anymore, should be removed. And that those cells are called senescent cells. And there are people trying to make medicines that are called senolytic medicines that are trying to selectively remove these cells that accrue with age in your body. So, in fact, there are people that are trying to remove cells that are dead weight essentially out of your body in the hopes that that'll improve what they call it the health span. Now, these cells are also important in wound healing. They have a particular role in other processes that they kind of convert over to. So it's never as simple as that. And the companies that have tried this have failed in making a successful medicine. They've went into clinical trials and it failed. So it's probably a more complicated story, like everything else in biology is. But yeah, there are people that are looking to alter the amount of and type of cells in the body to help you live a healthier, longer life. Especially the uber rich are very interested in living longer, it seems like.
Well, so I mean, it is interesting because our cells are, they die and new ones are born. So there's this constant regeneration process that's going on. And it sounds like, so the senescent cells are ones that are not dividing anymore. So they're not contributing to the death and the rebirth. They're there, they're not dying, they're not essentially reproducing, and they are hanging out. But you're saying that they also have some role in wound healing?
Yes, they do have a particular set of signals they send out that helps in wound healing, in recruiting cells to the area of a wound. And if you remove those senolytic cells, those signals are diminished and the wound takes longer to heal.
Okay, interesting. Yeah, and the senescent cells, because I love talking about fasting, is that one of the therapies that scientists are looking at for the reduction of senescent cells?
Well, no. What fasting is trying to do is prevent senescence from occurring in the first place. So there's several triggers that can trigger senescence, and one of them is an accumulation of bad proteins and bad things. So bad housekeeping within the cell. If the cell's housekeeping mechanisms are not doing well and it accrues a lot of damage to the cell, one of the possibilities, of course, to commit suicide is the cell. So you can go into apoptosis, and there's pyroptosis and ferroptosis, and there's different versions of that even. But the one is to just get rid of yourself. The other is to go to senescence. And what the fasting does is, in theory, it helps in improving the housekeeping part of it. So you don't have to go to senescence. So it improves something called mitophagy, which is essentially the overturn of mitochondria within the cell. So if you get bad mitochondria building up in the cell, one of the outputs could be senescence. But exercise and dieting and fasting, in theory, improves the cleaning up of the cell's environment, so it builds up less toxins. So it avoids senescence. It doesn't kill senescent cells.
Okay, so fasting cannot kill senescent cells?
Not that I know of. Never heard of that research, but there's always exceptions in biology. It's complicated. I've never heard of that research. I've heard that it avoids senescence. It helps improve the cell's kind of house cleaning that would avoid the senescence in the beginning, but I've not heard that it actually will remove senescent cells. I don't think it's senolytic.
Okay, but it could prevent you from getting more senescent cells.
Yes.
Yeah. Okay, got it. All right. So how would you explain to a 5-year-old how drugs are made today?
Oh, boy. Well, so that's a good question. I mean, it should, and it almost does always start with patients and what are the needs. Okay, I'm going to give you my idealized view. There is an idealized view, then there's also a cynical view. I'll give you, as a scientist, how it should happen. And it should definitely start with the patients and what people need.
You've already lost the 5-year-old. You've already lost the 5-year-old. No, I'm just kidding. Sorry, go ahead.
What you do is you want to fix people that are sick. So you try and figure out what's causing the illness, and you look at it, you try and mimic it. You try and make pretend versions of the illness. So you make pretend versions of the illness, and you see if you can make a medicine that fixes the pretend version of the illness. And then you see if that medicine that you made that fixes the pretend version of the illness works in real patients on the actual illness. So that's kind of how it's done. You make a pretend version, and of course, this is where another cause of failure is that the pretend version does not recapitulate the actual version.
You know, Dave, when you were 5, you probably knew the meaning of recapitulate.
Oh.
I'm sure you did. I'm sure you did, because from what I hear, you're really good at playing Scrabble and know a lot of words.
Yeah, maybe. So yeah, the idea is that if you make this pretend version of the illness, will it work in the actual real illness? That misses the mark a lot of the times. That's not the case. And sometimes it's because the pretend version, you weren't smart enough in making the pretend version, or you missed something even in the timing of the pretend version. So you may make it, but in actual life, the timing is off, and that actually can have an effect as well. So you have to try and be really, really good with your pretend version to try and make it behave the same in the real person. And the more you understand about the sickness, the better your chances are of doing that. Now, if it's a genetic thing, that's a single point mutation genetic thing, then it's a very easy, and you fix that genetic problem and you fix the thing, that's fine. But many diseases are far more complicated than a simple point mutation. And so that's when, especially everything that has to do with mood or, depression, anything psychiatric in the brain is just hard. And it's so hard that many pharmaceutical companies have left the field because it's a graveyard of efforts that have failed. So, it turns out that if somebody's authoritative in a white coat, that probably works as well as most medicines anyway.
If you want to create a more receptive body, free from toxins, inflammation, and metabolic issues, would it be crazy for you to explore booking an Ayurvedic consultation with me? There's probably never been a more perfect time to do it. Every day you wait matters when it comes to your health. So if you're ready to get your body back in balance, I'll give you a 30-day plan to get you started. Go to heathergrish.com to book a consultation. That's Heather, GRZYCH.com. Well, it's kind of funny that you bring that up because this morning I was reading the book How to Win Friends and Influence People by Dale Carnegie. And there was something really funny that he said in the book. And this book was written a long time ago. But at the time, there was an understanding that you say like 50% of I think he referred to them as, I don't know if he referred to them as like insanities or mental diseases. I can't remember his specific wording. That 50% of them were caused that they could sort of attribute some physical structural issue, maybe a lesion or inflammation or something, right? They could attribute some sort of physical manifestation to the fact that this person has a mental disorder in some way. But there are 50% of them that they really just have no idea on a physical level why something is happening. And don't you kind of have to know what's happening on a physical level in order for the drug making methods that are used by pharmaceutical companies to work?
Well, two things. One is it's not a requirement of the FDA to know how your drug works. That is not a requirement. Just that it works and it's safe. That's all the only requirements. Most drug companies want to know how it works so they can make choices. And in fact, the way medicines are discovered go both ways. So some do not presume. So there's two kind of methodologies at work, two common methodologies at work in the drug industry to discover ones. One is called phenotypic screening, and the other is called like high throughput screening or more mechanistic screening. One presumes you know the mechanism, and one does not presume that. One, you're just looking for.
Could you specify what the mechanism is?
A mechanism be acting through a particular protein or something like that, or a pathway that you're specifically targeting. And with kind of stripped down version, you have a hypothesis that you believe you know what's causing this disease. You've distilled it down, and now you're going to screen against something in that distilled version to muck up the process and fix the disease. And the other way is you don't presume to know the process at all. But what you do have is a pretend system, a pretend model of the process. Let's say you want a good example. Let's say you want to increase sperm motility. You want to increase sperm motility. And what you do is your test is you set up a little sperm racetrack, and you have a microscope. And then you just cover the sperm with different drugs, different sperm, different drugs. You have a little race. And you find the drug that makes all the sperm equivalent to secretariat. Now you've got secretariat sperm. That's very different than saying, I know the exact motor I want to speed up in the sperm. And then you take the sperm apart and you try and find the motor, makes the flagellum spin faster. There are very different approaches. The one is just a sperm race. That's a phenotypic approach. And the other is, I know what will make the sperm go faster. And that's like tweaking the engine from the inside. So that would be the difference. But a sperm race would be, by the way, I just came up with a really good way to create higher motility sperm for those men out there. And the nice thing about a sperm race is that, whatever medicine you found, it gets in the sperm, it goes faster. So your chances of having it succeed are very high. It's in the proper cell type. It's a very good recapitulation of what needs to happen. The only thing now you have to worry about is now that you have the sperm race in a petri dish, is it toxic to your liver? The thing whatever you do, because you don't know how it's working in the sperm. You just know it's going faster. Now you need to figure out, is it poisonous? And then now let's say it kills liver cells or something, or it increases your heart rate, gives you a better chance for a heart attack. Now you got to figure out, okay, maybe now you want to find out, what is the thing it's working on inside the sperm, or modify it to reduce its toxicity. So you can go both ways. One is called target deconvolution. So once you have a typic assay like sperm speed, now you want to figure out what is it the thing in targeting in the sperm, and then go from there back and make it from a compound that you just discovered to an actual drug, which will now be safe to take in orally or hopefully via a cream.
I really like you explaining that because the sperm race sounds really fun. And maybe there's people here thinking about setting one of those up. And yeah, and doping their sperm, and making sure that that doping of their sperm doesn't affect other parts of their body that are.
Oh, the jokes we could talk about with my little.
It's really great. But what's also funny is that the example I always think of in the Ayurvedic pharmacopoeia of dravya guna, so the substances that have certain qualities that are ingested into a body or added to the skin of a body or smelled or ingested in some way to change processes inside the body. One of the classic, we use a lot of herbal medicines of whole plant herbs or roots or whatever, but one of the classic examples is this one natural substance that's used for male infertility. And it has a great story about how it was discovered. Since we're talking about drug discovery and like you're talking about how it happens in a lab, but the lore goes that monkeys were observed licking it from the rocks. And somebody observed these monkeys, licking it, and then they'd go and like hump all the females.
Oh, okay. That's a hit.
Well, it's interesting because
we call that a drug hit.
Because one of the things I know that we have fun talking about is, old school pharmacology versus new school pharmacology. And the old school pharmacology, which is people that you were typically from indigenous cultures of, anywhere on the planet at the time, would go out in nature and they would observe things and then they would try things and they would test things on their own bodies or on certain people's bodies and things like that. But now we don't really do that anymore. We have chemists that are kind of doing what you're talking about, which is trying to identify the specific active ingredient in a drug and isolating that and figuring out which particular protein that drug, that substance is going to change the function of, and whether or not it's actually doing that. It's a very different approach to how indigenous medicines have always been done. And I think a lot of people question which way is really more effective. What are your thoughts about that?
So traditional pharmacology is actually quite an old field. I think it started in Scotland in the 1700s, the first kind of study, and it started with studying with the indigenous plants. So it started with the physiological effect of plants at the very first. So that's how pharmacology started, out of indigenous medicine. It's funny because when you look at the timing of kind of where it started entering university settings, it does coincide with the Enlightenment. It seems to coincide with around the Enlightenment time, where more scientific rigor and less spiritualism was the sway in Europe. And traditional pharmacology really did start, I think, in the universities in Europe. And they definitely started from indigenous plants. And by the way, that current pharmacology, you talked about that. I mean, Viagra was discovered in a way they looked at a side effect, which was an erection to a heart medication. And they're like, oh, these old guys are getting erections. Maybe we should, remarket this drug as something different. So even today, there's still an awareness of what's going on in the body, and they're not blind to what's going on. And people still try these things and still try and understand what's happening to the person. And by the way, even the largest pharmaceutical companies have what's called a natural products efforts. Many, many still have what's called natural product efforts. And it is true that when you find things in nature, they tend to be far more complicated than what man can do regularly. So most drugs, most man-made medicines that a chemist makes in a lab, they have a bias to making things that are easy for them to make. And nature, with billions of years of evolution, can make really complicated things. But even the biggest pharmaceutical companies understand that and will look at nature. They still will try and distill out the key ingredient because they don't necessarily want the other things. I think I mentioned that foods, just as foods have poisons, like grapefruit, or grapefruit's poisoning an enzyme that detoxifies things inside your stomach. If you take an herb, it may have something else in there that could do the same thing as a grapefruit does. And the pharmaceutical company wants to make sure that they have some modicum of control. And I use control in quotes because obviously they're still not great at, it's complicated, and even the best and biggest are not great, because if they were, drug approval rates would be much higher than they are. So only 1 in 10 in the end, from the very beginning to the very end, only 1 in 10 succeed, which is one of the reasons why drugs cost so much. And if we had very, very good understanding of biology, it would be much higher than 1 in 10.
I really appreciate you explaining that. And actually, one of the reasons I like to interview you is that sometimes I'll have a client come to me that is on a huge list of drugs, and then we're talking about giving them herbs. And it's really challenging sometimes because we know after a while that's got to be a lot for the liver to handle, for someone to be on so much stuff. I mean, even just like eating frequently taxes the body, and then taking all this other stuff taxes the body even more. So I think it's really great for people to start to understand what is in what they're taking, what it actually does to their body. Just even though, a lot of it requires big brains like yours that understand how to do calculus and were really good in chemistry class and didn't get a C like me, but because I really, really, really hated chemistry class. But you also mentioned that it actually costs so much money to make drugs. And I was just curious, like, how much does it actually cost for a company to research whether a drug works?
Well, that's a good question. And it is a very hotly debated one in pharmacology in the field. And it depends on how you count costs. So if you were to look at including all the drug failures and the missed opportunities of a pharmaceutical company developing a drug, so I told you only 1 in 10 succeed. So that means they still have to pay for the 9 that failed. So if you're paying for the 9 that failed, the one that succeeded costs around, these days, they say around $2.5 billion. If you are talking about just the money, the dollar amounts that go in the outlay that don't include the failures in it, it's hundreds of millions of dollars of cash that they have to put out. Now, that is further partitioned into what's called preclinical studies and clinical studies. Preclinical studies, including figuring out how you're going to attack the disease in the first place. So the target, what's called target identification, and that can take years and years and years and years and years until they figure something out.
And that's like, what do we think's going wrong in the body that we need to actually change?
What do we think is going wrong? Yeah, if you're doing a non-phenotypic version, if you want to try and figure out a target to muck up, figuring that out can take years, and it can happen. You're reading literature, and there may be a buildup of time and literature and more evidence that's showing that something in this pathway needs to be fixed for this disease to be fixed. But that can take a lot of time in normal research. Sometimes it happens fast, but sometimes it can take a long, long time. I mean, we still don't know the cause of Alzheimer's. So all the theories of Alzheimer's that have been tested have failed the test. So, there's lots of theories, but they've all failed the test. So Alzheimer's is one. We're still learning what to do. And so that's one part. And then once you have an idea, and then it's fairly prescribed what gets done in the preclinical thing before it goes into man. And then once it gets into man, there's 3 phases. Some of them can be broken down into sub phases.
Hold on, you said once it gets into man, like person?
A person, yeah. A man or woman.
Okay, I just wanted to make sure you didn't say in demand, but you said
No.
essentially once we actually test this out on a person.
Yeah, in a person, yeah. So it starts out at a few healthy people that don't have the disease and seeing if they're going to, how bad it is. Then you're using very small doses. You're not actually trying to affect anything other than how toxic to determine if it's safe to put in a person. You need a few people, and you're looking at the metabolites and how the things being broken down in a real person. And if there's a drug-drug interactions, that's maybe when you find out that, this person ate grapefruit and all of a sudden his plasma levels of this drug shot up through the roof. And then you find out, you ask the person, your plasma levels are super high. What did you have for breakfast? Then you can find out things about what's going on with the medicine. Phase 2, now you're looking at actual people with the disease that you're trying to fix. Now, the problem happens here. One of the things that makes it very expensive is trying to recruit patients in. And that's a science in and of itself. But also imagine a situation where you've got 5 drug companies that are all trying to affect the same disease, let's say lung cancer. And now you've got 5 drug companies that have medicines ready to go into trial. They're now competing against each other to recruit patients, right? And that could be expensive and time consuming. So now, let's say there's only several thousand patients or whatever available, and some don't want to do it because they don't want to be on an experimental drug. Some want to do it. And now you got a subset of those people who go into a trial. So it's really complicated and take a long time to just get the people to submit to a clinical trial. And then the phase 3 clinical trial is in a much expanded trial. And now you're trying to get real statistics. And is your drug efficacious or not?
That is a lot of work and
Yeah.
so much harder than me just going, okay, left quadricep, do what I want you to do.
Yeah. It's very expensive. And the phase 3 clinical trials, now you're recruiting, it could be 1,000 people. Those doctors have to get paid. Those trials can cost over $100 million, depending on if you're going at a disease like high cholesterol, you may have to have 10,000 people in your trial. And that's a very, very, or Alzheimer's, where you have to do it for a very long time. Those trials can cost, some trials cost several $100 million. So it's hard.
You mentioned the natural products departments of the pharmaceutical companies. And I guess, let's just use would somebody study cinnamon in the same way that they would study something that was synthetically developed in a lab?
The answer is absolutely yes.
And who's studying that?
Well, if there were an interest in it. So, for example, I believe in Ayurveda, cinnamon would be like a blood thinner. Is that correct?
Yes, it's warming. It stimulates your digestive power, your digestive fire. Yeah, liquefies.
Okay. So what you do is, if it was a blood thinner, and that's a very important, there are drugs that are used as blood thinners. Some of them are dangerous. So yeah, if you could stick cinnamon and test it to see if it thinned blood, which is not a hard thing to test, then they would try and distill the elements out of cinnamon. They would separate the components of cinnamon to find out what is the chemical in cinnamon that's thinning the blood. And then they would say, is it safe to give on its own in the doses that you would need to be clinically relevant? And if it's not, could we modify it slightly? But the advantage of the thing that natural products like herbs have is they're much more three-dimensional than most medicines that man makes are. Most medicines that man makes are more two-dimensional. They're more planar. They're not so complicated.
Can you explain what that means?
Literally, they are more simple looking. If you were to look at a natural product from a fungus or from some other plant, if you were to just look at how three-dimensional it looks, they're much more complicated things than a chemist would ever even dare try. In fact, there's a whole field of organic chemistry to try and see how elegant your chemistry is, to try and recapitulate what a plant did super easily. But you can get PhDs by trying to synthesize something that was done in nature. So there's whole fields of chemistry.
I think that's a really interesting point that a lot of people don't really think about, is that what I hear you saying is that what nature has made actually is more complicated than what man makes, though my brain cannot understand half the stuff you're talking about.
No. It just looks more complicated if you were to see it.
But frankly, I couldn't really understand how nature would make things either with that similar kind of intelligence that you're talking about. So, you know what? This has been so fun, and we'll do it again soon. And I'm really happy that we have this second interview, because like I mentioned earlier, language is sometimes difficult, and I wanted to make sure I got a better understanding of what you meant by the idea of poisoning a body, which I don't think is really what you meant, but it's the sort of technicality of the word. But thank you so much for being here, and I think everybody probably got a lot out of this today, so thanks so much.
All right, no problem. Thanks for having me.
Thank you for tuning in and dropping in with us today. Don't forget to subscribe, rate, and review this podcast. I really want to hear your feedback. To learn more about my work, visit heathergrish.com. That's Heather, GRZYCH.com. And meet me here next time on The Wisdom of the Body.